Thank you very much. In order not to delay things, maybe we should just start the roundtable discussion. I just want to remind everybody, if they don't mind, just filling out the questionnaire, which is in the chat. And thank you very much for all of you participants, the panelists, for being here. If you want to open your cameras, then we can see you all. And we'll try to go in order. I don't know if to just tackle first the questions that were left from the previous sessions, which were, I think, majority, well, there were some related to serine, but there were other questions too about structure. And so I'm just gonna start hitting, and Mela, please interrupt me if I forgot the question or something, and feel free. There were some families asking how long would their child would need to take L-serine for it to have an effect, and for how long is it for life?
Well, according to the experience that we have, the positive effects in some of the aspects of neurodevelopment for most children are seen quite fast. I mean, in the first couple 2-3 months after starting taking serine, it is possible to see already some kind of improvement in different aspects. And anyway, we have our experience is just very initial, so we have little experience. That's why we are now conducting the clinical trial, just to have more patients and see how is the pattern. It is true that probably because it is not only the action of L-serine into the NMDA receptor to activate it, but also recomposition of metabolism, because L-serine is also a precursor of sphingolipids for neuronal membranes, that it also participates in a particular energetic pathway and so on. It is possible that for serine for a long time has other effects that we still don't know, that can improve also other functions and be beneficial for a long time. If for a patient has a positive effect, we don't have any reason to remove L-serine. It is the same situation for other metabolic disorders or other conditions where L-serine can be administered forever. I mean, it doesn't have negative effects. So yeah, if this has a positive effect, it can be given forever.
This question is kind of for both of you, Amy and Angels. This is, are there any L-serine testing in mice, undergoing in mice?
Yes. So we've been chatting in other, we've been having discussions about what would be the right dose. Chavi's done work in mice, haven't you? Chavi, done some L-serine treatments in mice?
Well, not yet.
Okay.
We're waiting for the approval from the ethics committee.
Okay, great.
Hopefully
So,
we'll start in January 2021.
Okay. Sorry, I misunderstood then. So we've had mice on the diet for 2 months. We've had the knockdown mice on the diet for 2 months, and we are going to start testing this week. So I wish that I had some answers for you, but so far so good. They're doing fine. No one's looks worse, but we haven't done the tests yet. So I will be happy to convey this to however however would be best. I'll spread the news.
Thank you.
Just a comment that is important, and because of the half-life of L-serine, it is important that L-serine has to be given at least 3 times a day. So with because of the half-life of L-serine, that drops the kinetics on blood, and if you don't take L-serine for more than 6 hours or so, you don't have the proper effect. So if you take all the necessary dose in a day, but just once a day, it doesn't have the same effect that if you divide the doses of the total day at least in 3 doses. 3 or 4, better 4 doses will be more recommended. There is a very recent paper by Carlos Ferreira explaining that, because L-serine is given now more and more for different situations, clinical situations. So this is like an important message. We just, in our protocol for the clinical trial, it is like that, but just, I say that just in case somebody is taking L-serine in an independent way. So this is an important message.
I have also a question about the L-serine trial. There's a family asking if you have a placebo arm in the clinical trial. So people who are not taking and you're comparing side by side.
No, this is probably, this is the best way to do a clinical trial, but the problem is that we don't have too much patients. We have just the sample of patients is small. So if we do that, finally we will end up with very, very few patients, and the results will not be strong enough or so robust enough. So we have tested like development or the normal pattern of development for some time before L-serine, and we compare in the same child, what is the evolution after taking L-serine compared to the normal pattern of development before L-serine. But we don't have enough patients to do this placebo. It would have been the best situation, but no.
Thank you. Now we move to something else. How much is known about the expression of different subunits and different cell types? So I guess that maybe for Dr. Wen. So the expression of GRIN in different cell types and the different subunits.
So we haven't tested that in all the cell types yet, but definitely in the neurons that differentiate from the iPS cells, we did the Western blot before that we can see that NR1, NR2B, and NR2D are expressed, and yes, but we haven't tested that in other cell types yet.
I have an interesting thing. I didn't include this because it was very technical, but it might be interesting to show. Can I share my screen for just a second? What this is in the mouse. Can you see this? Okay, so this is, similar. It looks similar to what Jay Singh showed for the brain organoid, but this was done with 20 organs from the mouse. And so on the left, you can see all the different cell types in the mouse. And these are not different organs, these are cell types. So for example, these are all adipose tissue. This is all adipose cells that are found in the many different organs. And then on this side over here, what I have, I asked to look for the expression of GRIN1. So this is the expression of NMDA receptors, or at least GRIN1, in all the different cells. And so you see, of course, that they're brightest in neurons, but in the beta cells of the pancreas, they're also pretty bright. This is epithelial cells here. These are basal cells in the tongue, in adipose tissue, and these are T cells here. These are microglia here. These are astrocytes here. So I guess what I wanted to show you is that even though they are certainly highest expressed in neurons, they are also found in other cells of the body. Oh, these are your B cells here. These are the progenitor B cells, and these are the mature B cells. So the idea that they could be expressed on immune cells is, at least in mice, there's evidence. Now, this kind of technique is not as easy to do in humans, in postmortem tissue as it is in mice, but there is certainly evidence that it's expressed in other cell types besides the brain.
Yeah, thank you, Amy. Actually, I want to say that also we did the single cell RNA-seq for the human brain organoids, and we can put out the data for all the subunits to see their expression in different cell time. Yeah,
that would be interesting.
Interesting compare. I'm sorry, Christian just pointed out that there were a few more questions for the serine that I didn't address, and one of them, maybe I'll lump them up. What are the foods that contain serine? I know Natalia briefly mentioned them. And if you don't know gain of function and loss of function, would you still profit from taking L-serine, or is it better to get a proper functional diagnosis? Where to get L-serine? It's expensive, etc. And if, again, going back to if it's only used on loss of function. So.
Yeah, but according to the foods that are rich in L-serine, there are many of them. You can probably find it better quantified in web pages that are and applications for diets that have exactly the content in grams, etc. So, but I think one of the most rich foods in L-serine is some kind of meats like turkey, also eggs, soy proteins. I mean, these are some of the most enriched, but then there are several other foods that you can easily find in web pages that quantify different amino acids and other components of the food in every kind of product. I mean, this is very easy to find. But this is one. Then yes, L-serine should be theoretically used in loss of function. We don't have the rationale for giving that in gain of function. Well, it is not established. I mean, it is just to activate the receptors that doesn't work properly, that are that work quite slow and worse. So if you give L-serine in gain of function, you won't have the necessary effect. Maybe you have other effects like those that I already mentioned, or the remodelation of lipids of membranes and the energy aspects, but that we don't know. We just don't know. So it is preferred to, first of all, to know if it is a gain of function or a loss of function before starting. And then the other question was where can be found L-serine in the internet, or there are several sites where we can find it. First of all, Nutricia, you have the webpage from Nutricia. I don't know if you as a parent or as any person can buy it independently from a hospital. We just ask the dietitians in our hospital to send a Nutricia, the L-serine from Nutricia to every patient, and this is for free in Spain. But well, I don't know how does it work in other countries. I think that I guess in other countries it is not the same, or at least not in all countries. It's like that, like a prescription, like a medical prescription. Otherwise, maybe a person as an individual, a patient, can also ask a Nutricia for L-serine to receive that at home. But then there are other sites, other trademarks that have also apparently a high purity, and that are, they can be administered without any problem, because it seems that, well, the purity is high. We have I think a web page that we can share later on if you want, Sandra, that was given also to us from other researchers, that maybe, well, it is also interesting for patients.
Yeah, and I think maybe we should add again, if you are going to take the serine, talk to your neurologist. If you need your neurologist to get in touch with you guys, we can, provide you with the email contact, because I think this is the best way to do it. And then they can monitor your child accordingly. If you want, you can do previous testing, kind of what you're doing in the clinical trial. And you are getting some patients, I know, from other countries that want to join. And basically what they do is just follow the protocol of the clinical trial. So I think that's also important. So I think hopefully we covered all the serine ones so far. Now I'll just go. So there was a question also about the symptoms, and if you see the same symptoms with the same protein change, or I should say the same amino acid change. So when you have a missense mutation, if it's in the same position, would you see basically the same symptoms?
Is the same symptoms for the same mutations all the time? Well, it doesn't happen normally in any other kind of genetic disease as well. Sometimes, yes, you have a correlation between the genotype and the phenotype in some occasions, but this is not something that happens always. This is not a constant. Yeah, it can vary.
This is a point that I would have. Sorry, please go ahead, Mireia.
It's just that what we see is that there are some important positions, some positions that are really conserved and are essential positions. And then when these positions are the mutated one, then usually the phenotype is more severe, and then we have seen some really slight changes in the physicochemical properties of the amino acids, and in some positions that are not really important. And then in this case, the phenotype seems that to be more mild, less severe. But it's just a general observation. This is why we need really a patient registry and to know all the information from the child, and then we will be sure to make a correlation of severity and to the clinical symptom related to some positions in the protein and to some amino acid changes.
Just a point related to that. It's quite possible, likely, that other mutations, other polymorphisms, not disease-defining polymorphisms across the rest of the genome, will be modulating the symptoms of GRIN mutation. So other genes that are slightly different from people to people may enhance or weaken the effect of a GRIN mutation. In that context, Shashing, would it be, what do you think? Is it better to isolate the mutations and use one backbone cell line to put in your different GRIN mutations and grow 384 brains at the time with different GRINs, but all in the same backbone? Or do you need to look at IPS from patients?
We'll use both. We'll use both. So we already generate the patient IP cell line, but next step, we will do the isogenic line by introducing the same mutation to a healthy and to a control backbone. But on the other hand, we also will correct the mutation in the case iPS cells to convert to the wild type. So we will have two different type of isogenic line. One is the correction, and one is the we call the knock-in.
Yeah. Okay.
I think I would second that. Just that we're all more than one single gene, and what makes us who we are is not whether our status at just the one gene clearly. So how that expresses is going to be different depending on your own unique genotype and everything that makes you you. And we see this in the mice too. The mice, we have them all on they are all identical twins. So they're all on the same genetic background, but we can move that mutation onto different genetic backgrounds. And we see that the phenotype of the mice can become worse or can become better depending on which genetic background we use. And so we have to use actually a special genetic background in order to get the mice, the knockdown mice, to thrive and to to live. If we put them on certain genetic backgrounds, they don't live. And if we put them on other genetic backgrounds, they're even more robust. So we've already seen this in mice. So it wouldn't surprise me at all that depending on all of your other genes, you would have a more or less severe phenotype.
Yeah, and I think your data, what you showed in the mice with the various tests, actually I was surprised how diverse they performed. The standard deviation in your columns was quite amazing, given that these are identical twins, one to the other. Yet they perform quite differently now, even in that setting.
Yes, I imagine for an immunologist that data probably looks really dirty, but yeah, but that's that's the thing, I wish I had a blood test. I wish I had, I wish I could do what you do, because yes, the mice, depending on whether they got in a fight that day or whether they just ate or whether they've been, we don't know all the things that make them choose to behave a certain way. But it's not just that, it's not just even their genetic background. Yeah.
It's also impacts our kids, to exact that degree, whether they have eaten well in the morning or slept well at night.
Exactly.
Yeah. Okay, thank you.
Exactly.
We have a question about binding. So if you have a mutation on the binding side of glutamate and you use an agonist, would you actually see a benefit? Because of course you would only act on the healthy receptor, which you might already have decreased because you don't have the binding site. So I don't know if anybody wants to take this one.
So the question is about those mutations that affect the glutamate binding site, and that there is an absence of binding. But I think the strategy is focusing on the wild type subunit that is still present in some part of the receptors, in a pool of the receptors. So this is not affected by the variant. Only those that have the mutation will be unable to bind glutamate. So we will focus on those that are working properly, and then the idea is to potentiate those that are responding to glutamate. The other ones, of course, will be not responsive.
I think just to clarify that question, I think that the question was more because I know it's known that if you have a mutation on the binding site, and that's the issue with the whole NMDA receptor, that you will focus on the functioning one. But are there like any approaches, or are there any work being done on the dysfunctional receptor where the mutation causes the inability to bind glutamate? Okay,
now I get the question. So trying to act directly on the mutant, this is pretty difficult. So we and pharmacologists have a lot of difficulties to develop new compounds that are having a beneficial effect on NMDA receptors. So we can imagine that designing new compounds that will interact with these mutant receptors that have an affected glutamate binding site is very, very complicated. It means a lot of effort for a single variant. And since there are many different variants, it is almost it's very challenging. So I think that most of the people are focusing just in a set of receptors, the wild types.
Okay, so just to understand this correctly, does that mean that, because how I understand it, that different variants can cause the same dysfunction on the NMDA receptor? So there are different variants that cause the inability to bind glutamate. So if we assume that activation of an NMDA receptor requires glutamate binding, there will be a group of variants that will have the same issue, right? So it's not specific variants, but a group of variants that will have the same issue. So this is merely so I know that it's difficult. I'm just interested in knowing whether there is any work being done in that, because at all, as I understand it, there there are quite a few variants that actually cause inability to bind glutamate. So if the overall approach for kind of this type of mutation is simply just focus on enhancing the normal copy and not so much focusing on fixing the bad one.
Yes, I think that this is the idea, because even if they are affecting the same pocket, the glutamate binding domain, in terms of the molecular, the amino acids that are affected and the changes in the structure, they are pretty different. So you are affecting the same domain, but the amino acids that are affected are different. And then if you develop a drug, this is there is a very tight interaction. So it's not any kind of compound will not bind to the different mutations affecting the same domain. So perhaps, Mireia, you want to add something in terms of structure and?
Oh yeah, but I think the same way. So yes, for sure there will be, I guess, in the future, maybe the groups of variants. So looking at loss of function variants, we have those that are avoiding the subunits interactions. There will be some of them that will be related to avoiding glutamate binding. Some of them will be in the transmembrane region. Some of them will be just really at the bottom of the receptor. And then maybe in this case, if we have in the future more available drugs, and these drugs are binding in different regions, we will be able to choose different drugs according to the domain where the mutation is found. But the other side, I think that if we have a mutation that is affecting the glutamate binding site, then probably working with positive allosteric modulators that work at the top of the receptor, we'll also have some problems. So I guess for those that are glutamate binding, the best thing is just to work on the wild type copy. But maybe there will be some drugs in the future that will act in different regions. We could even certify more the different variants, the different loss of function variants. But right now we don't know if we can rescue the mutant receptor function for those that are avoiding the binding of glutamate.
I guess I just wanted to add about drug discovery. I mean, there's kind of two ways you can approach it. One is a very rational one where you have the crystal structure and you can kind of fit in and try different drugs and predict that they're going to do something. But in general, we usually use screens where we're just looking for some signal that something has improved. And so it could be that when we do these screens, I think Jae-Sing, lots of people are developing platforms to screen new drugs. There may be one that ends up working that way. And I guess this is why screening on several different variants will be valuable to see whether it works for all of them or it only works for one of them. But it probably won't be a rational kind of, prediction, although there are some people who are doing that. And I guess GRIN is trying to pull in scientists from all different disciplines so that maybe there will be someone who will do that. But in general, the drug screens are kind of more just looking for a benefit and then seeing how broadly it could benefit different variants.
Yeah, and that makes a perfect, that makes sense, of course. But it was just one of the thoughts that I had. If the variant kind of is within that section where it actually affects the glutamate binding, so the purpose of looking for a drug that could give you a benefit, it wouldn't make sense to look at the dysfunctional, receptor. Then you should focus on the enhancement of the normal one. So maybe that's I don't know how many variants actually do that other than my sons, but I know that a few do, a few of them do. And I think that a lot of the parents, even though they have an understanding of, you know, the function of the NMDA receptor, they don't actually understand what it means if their child has a variant that has, you know, that affects the glutamate binding site. So that, I think, hence all the questions about, you know, would the L-serine work if my child, you know, so it's also more of a general question for you, so that you can explain for all of the parents, you know, how all of it works, and maybe that group of GRIN patients, you know, they might be the most difficult ones to actually find a treatment for.
Okay, we continue. I don't know, Amelia, you want to ask about the mice? You also mentioned something about the mouse models of the binding.
No? Oh, that wasn't my question. It was just a question from, I don't remember who. I just.
Yeah. No, there was somebody also asking if there were any mouse models of the with the binding, the glutamate binding defect. And I don't know if that's the case of the GRIN 2B in specific, but I don't know if you know of any of the other ones, because I know you don't have any, Amy, but I don't know if you know of any of the other ones.
I think there are some being developed. Keith might know actually better than me. So the GRIN ones are mainly the ones I'm doing. So that's for the, you know, that would be more the glycine or serine site, but then for the GRIN 2A or B, the glutamate, we have gain and loss of function, more loss than gain. I know that people are developing, so that list is now out of date, and there are a lot more that have been developed. So we need to make a new list and find out. I think there are more mice even than that list shows. So I think there's a good chance that one of them will be a loss of function on either 2A or 2B, probably 2B, just because it seems like there's more mice being made of 2B than 2A.
I have another question about gene editing, actually. The gene editing being considered in as a treatment for GRIN gene?
Shay-Sing, are you doing anything with your organoids?
We just use that as a tool for the research, but you're talking about the gene therapy. So I think there's a couple of things we need to think about. One is the how to deliver the gene. I mean, right now we see, I mean, for other disease or for other cases, people trying to use AAV, a different AAV or other kind of virus. And I think we first need to test how efficient, what's the efficiency for the delivery, and where there's other side effect or toxicity, and also need to figure out what's the best time point, what's the for the gene therapy. And I think to do that, maybe need to do some preclinical study before moving forward to the patient. For example, use the animal models or use the primate models for test that out.
Yes, if I can add something. So we heard a lot of these promising CRISPR and gene editing strategies, and few editions before of this symposia. We also heard some presentations. So there are a lot of hopes, and social media are very, they are stating that this is like the new medicine. But to be, well, this is my opinion, but to be realistic, thinking about gene editing in vivo in children for neurological conditions, it is perhaps well, too early to think about this. So right now, I think that the more realistic options, and well, the physicians will correct me, but are those that have been presented as those compounds that are already in the market that can be repurposed, and also tackling different aspects of GRIN disorder symptoms, as Jenny Lee nicely presented. So there are many aspects that can be addressed in inflammatory or bioenergetics, or well, as well as the ketogenic diet that Amy presented. And these are the real, well, to me, these are the real points that need to be explored and to see really which are the benefits.
I'm going to answer a few more questions here. Oh, sorry, not me, you. I'm going to read them. So this is for Amy. Do you know ester protein glycosylation is altered in green knockout? Have you looked at it?
Sorry. No, I don't have any information on that. There is a new investigator joining our department who specializes in protein glycosylation and all those enzymes that do that. So maybe we can find out something about that. But at this point, I have no evidence that there's any change there.
We have another question for Dr. Ramsey, and this is, I know that Xavi and Dr. Garcia mentioned a few chemicals that they were trying, or at least they were studying for children, and they were wondering if you actually have tried something similar. I guess there's some numbering, and including the L-serine, which you already answered, and the piracetam. If you have tried that on your mice.
No, I haven't. And I mean, we had a meeting where Dr. Garcia Cazola said that we could consider adding spermidine in, spermidine or spermine, I can't remember which one it was. But the only one that we've done so far is the L-serine, and I haven't tried any other. So the other medicines that we're trying right now is CBD, cannabidiol, and THC, and combinations of those. And we're going to be feeding them cookie dough every day for 2 weeks, and then testing them in seizure and in you know, some cognitive tests, and also look at their sleep patterns. So that's I would say that the L-serine and the cannabinoids are the ones that we're looking at right now, but there are so many that we could and should be testing, and I think that having, you know, this is where the parent organizations can help inform us and let us know what are the priorities for the patients. What are you most concerned about that we could attack and focus on those things, so that we're using our time the best way we can.
Perfect, thank you. There's another question for you also. Can we consider BHB and ketogenic diet not only for GRIN 1 but only GRIN 2?
Yes, I don't see any reason why that would be limited to GRIN 1, because it's not, it has nothing to do directly with the GRIN 1. So I would say it would be more appropriate. It would, at this point, we've only looked at it in loss of function kind of scenarios, but I have no information about gain of function scenarios. So I would say that would be the more, I don't have any answer about gain of function, but at this point we're testing it in loss of function scenarios, but I don't think that it would be limited to GRIN 1.
Another question for Dr. Ramsey. GRIN 1, that you're testing on GRIN 1 mice, you see a higher locomotor activity, but you talk about energetic deficits. How do you explain that?
Right. So, well, actually, I think there's a bit of a conundrum there, because I think that when we look at brain activity and we look at neuron firing, the neurons are actually firing more than less. And this might be one area where it's different than what Jae-Sing sees in the organoid, where they're getting beautiful nutrition, and they're getting everything that they need. They're bathed in this constant, perfect environment, right? But the neurons are firing more. But if there's a problem with energetics, then that's going to have more long-term problems in terms of the neurons being able to maintain their ability to fire and their ability to carry out all of their different tasks, that they're going to run out of energy, because the more a neuron fires, that's very energetically demanding. And so more firing means it needs more energy. And if it can't get the energy, then ultimately there's going to be a failure, and that could be the neuron dying or not working. And so the more activity is probably more firing of certain neurons. But I think that's the bioenergetic deficit would be something that we'd see more long-term in terms of neurons failing and dying. And I don't think that would be reflected in the running behavior.
We have a few more questions. There was a question about the association between hyperactivity and gain of function. If anybody wants to comment on it.
I have one mouse that's a gain of function mouse, and it does not have the same hyperactivity. So we saw a mild hyperactivity in the juvenile mouse. It went away at when they became adults. And then when we tested them at 1 year, it looks like there is some hyperactivity, which, you know, they're also having more seizures at 1 year. So this was with the gain of function mice. So they do seem to be different in many domains. So the hyperactivity is one where it seems like that they're different.
Can I add to this? So of course I've only been studying the acute effects of inhibiting the receptor, but as I showed, there is hyperactivity when you block the NMDA receptor acutely. And also we've seen increased pyramidal cell firing when giving ketamine or PCP to rats. So the acute effect of inhibiting the NMDA receptor is actually hyperactivity, which is a bit thought-provoking.
Yeah, that actually might be a question here for you too, Chartan. It's some parents have actually seen the kids experience low sensitivity to anesthetics, and you know, could this be explained by the NMDA receptor? And I think also some of them, I would add to this, that even the pain, you know, sensation seems to be also out there in most of these children.
Yes, so I'm not a doctor, but based on the mechanisms, it makes sense. Many anesthetics inhibit the NMDA receptor, and ketamine is also used for pain treatment. So that could definitely play a role in pain sensation.
Angels, have you seen? Oh, sorry.
No, I just I always have to add that what is giving the symptoms is also to a large degree the compensation of the loss of function, I think. So as we also have seen today, there's so many glutamate receptors, so they can somehow compensate. And this may also be part of the disease you see.
I just want to add that I also, it was mentioned in one of the chat groups that some of the kids after the anesthetics had like a period of being very calm, very relaxed, like a reboot reset. I think we spoke with Amel about it, you know, like you're resetting the craziness that's going on in the brain.
Yes, so this is a well-known phenomenon with ketamine and also other anesthetics that you may get like a feeling of high when you wake up, and it's, I mean, but this is based on this acute inhibition of the NMDA receptors of potentially other systems as well.
Thank you. There are families that have seen deletion mutations that lead to specific phenotypes, like a more milder form of disease, and a milder form of autism and hyperactivity. Is this also seen in mice? I guess that's for Amy.
Yeah, so there was a paper published, I want to say in 2000, and the first author is spelled Kew, and they did something where they had point mutations, and they also had just straight knockout. And it did seem that the point mutation had a more severe phenotype than the knockout. So I think that that would be consistent with the idea that it was actually, no, I'm sorry, I'm saying this wrong. So a heterozygote with a point mutation was had a more severe phenotype than a heterozygote with a null mutation. So the null would be like the deletion, and so you know, probably your kids just have one version of the gene has a variant and the other version is normal, and so that would be what we would call a heterozygote. And so yes, there is some evidence for that in mice, that actually these point mutations are acting in what we would call a dominant negative fashion, that they're having an effect on they have an oversized effect on, you know, it's not like you would have half of the receptor. And I think it's because they combine together as a complex, that, you know, just having one, you know, misfunctioning subunit is enough to affect the whole group, right? So really, in order to get a functioning receptor, you need, you know, all of those subunits to be working properly.
Thank you. I'm going to combine these two questions about serine. One of them is if you have already normal levels of serine in the blood, or I guess in this case will be the CSF, is there a reason to supplement? And I will combine it with how is the dosage determined for L-serine, and I would assume are the papers and the medical literature, and what is piracetam?
Mm-hmm. Yeah, all our patients have normal levels of L-serine because there is no problem in the biosynthesis, in the production of L-serine by the body, or so this is always normal. There are some rare disorders, metabolic disorders, that have this impairment in L-serine biosynthesis, and in these patients we find a depletion of L-serine in plasma and in the CSF. In GRIN patients have normal levels of all kind of amino acids. There is no problem of synthesis, but still we give a more quantity, well, a high quantity of L-serine because we want to obtain this agonist effect on the NMDA receptor. So we can monitor for sure the L-serine levels after starting treatment. This is not strictly necessary, but we can monitor that just in order to see the treatment is performed correctly according to the recommendations, and also for research purposes if we want. But yeah, even with normal levels of L-serine, yeah, we'll give that because we want to have this action of potentiating the receptor. So this was the question, and then the other question was Sandra was another question about piracetam.
Yes, what is piracetam?
Was it? Yeah, piracetam is not repeal. Is like a cognitive enhancer. So I just will explain a little bit in the talk that it potentiates different types of neurotransmissions. So not only a glutamatergic transmission, but also dopaminergic. GABAergic is a molecule that is similar to GABA, and it potentiates the binding sites of NMDA receptors. So it will increase the possibility of glutamate or L-serine to bind to the receptor. And this is something that is given in pediatrics. We normally give that to treat myoclonus, which is an abnormal movement, non-epileptic myoclonus, and it works. and beyond neuropediatrics, in adult neurology, it is also given to patients with some forms of dementia or cognitive problems to enhance cognition, and also as a stimulant. Some students take that to be better at when they have tests, when they have exams. So this is no triple paracetamol.
Thank you very much. Thank you to all of you for joining us. Now we're going to hear a few words from our sponsors, GRIN. So I'll give it to you, Keith, to do the next couple of minutes of presentation, and then we're going to have just closing remarks at the end. Remind you that if you could fill out, please, the survey in the chat so we know where you're coming from. And we want to thank Marita for the video that you saw during lunchtime, and we have a chance to show it afterwards. It'll be great. Thank you very much.
Okay, so thanks so much. And I want to just start by thanking GRIN2B Europe Association for putting this together. The sessions that I saw were just amazing. Thanks to all of the researchers. I didn't get a chance to see everything yet, but I look forward to seeing the rest over the next couple of days. The next thing, I just wanted to give a quick news update. I know that there's people here from all around the world, but I think we've all been losing priority over the last few days. We've been waiting to see what's been happening in the US, and over the last 15 minutes, CNN has called the election for Joe Biden. So I think we'll be able to return to our productivity now. I'm in Canada, but you know, we're still obsessed over what's going on here. It's amazing. So I wanted to follow up on something that Amy had mentioned, which is that the associations need to help define the researchers' priorities. And we're kicking off a process now, and I just wanted to talk for a minute about how you can get involved. So the traditional model that rare disease associations use is that they begin with a request for proposals, and then researchers come forward and suggest ideas, and then the associations hand out funds to the project that sounds the most worthwhile. What we're doing is a little bit of a different process. This is based on something that a physician who leads a rare disease association has come up with. It's called, his name is David Fajgenbaum. And so it's the Fajgenbaum model. And basically in this model, we start by consulting with families, researchers, and clinicians to identify the priorities, and then go out and find the best researchers to answer these top questions. So here's kind of our process for doing it. So we've begun a research review where we are kind of summarizing all the work around GRIN that's been published already or that we know is in progress in labs around the world. The next step is a consultation period with parents, researchers, and clinicians. And the main reason I wanted to highlight this today is because over the next, just before the end of the month, we're going to be launching a survey where we want to get input from as many GRIN families as possible. This is really, your opportunity to help define GRIN research over the next few years. So we're going to be asking questions, for example, like how should we balance our focus on, kind of short-term interventions that might only help with symptoms versus long-term cures that could have a wider impact. And then we're going to be asking, how you would prioritize the concerns that you have about your child. Is it more important to you to deal with seizures, or to deal with visual impairment, or to deal with speech impairment? So we're going to be putting this all together, getting your feedback, and then we're doing another process with researchers and clinicians to get involved in this as well. And then we are going to be defining our research priorities, and then the final step is to go out and recruit the best researcher for each priority. So it may involve more requests for proposals, but it may be just knowing that there's one person who can do this work that we need answered, and going out and trying to get them involved. So the best way for you to make sure that you're involved is to fill out the survey, and the best way to make sure that you're getting the survey is to make sure that you're on CureGRIN's newsletter. So go to our page at curegrin.org, scroll down, and you can see that there's a sign up button at the bottom of the page for the newsletter. We've got about just over 200 families, I think, that are on there already, but we want to get as many families as we can before that launches. Where are the funds coming from for this? Well, we're doing a research fundraiser right now for research. We did one last November. We call it Count Me GRIN, and we sign up families to try to help us go out to their own families and friends. We're still looking for more families to help us with this campaign. It's called Count Me GRIN. So whether you're able to help us by being what we call a GRIN champion and asking your contacts to donate, or whether you are able to just make a donation on your own, every dollar helps. And this is really the funds that we'll use to help us prioritize that, once we have those priorities, these are the funds that we'll be able to use for that. And just one more thing that I wanted to mention, we have been putting together a book of GRIN stories that parents have submitted. And that book is also expected to go on pre-sale on Amazon at the end of this month. So it'll be both in paperback and ebook through Amazon. And so it'll make a great holiday season gift. So watch for that. And yeah, we're really excited about that. And I think that's everything. But thanks again to all the researchers and to the GRIN2B Association, Europe Association.
Okay, thank you very much, and thank you for your support. So we're trying to tag and unify our work as families. So I'm glad that we can join forces and try to get the opinion of all the families here and over there in North America. So I just one thing I wanted to ask you, please, besides filling out the survey, is we are trying to gather information about attending physicians for your children, because we try to build a consortium of neuropediatricians here in Europe that would better understand the disease of our kids, because we get a lot of families concerned that, they go to a neuropediatrician and it's very tough for them because there's very little information out there. So and it has shown to work really well in hopefully future clinical trials, but certainly in the Elserin clinical trial. So if you guys would like your neuropediatrician to join this group, please contact us at the GRIN2B on Facebook, via email, whatever way. We would really appreciate it. We will put up the video that you briefly showed during the lunch break that Marita made. We will have it ready and we'll hang it on our Facebook page for all of you to see, and you can review, and there are actually important links there. If you need the links, also they will be there for you to copy. So we will try to have like an actual copy of all the links next to the video publication. With that said, I just want to thank our panelists for spending a good part of your Saturday here with us, or basically your whole Saturday with us. And again, thank you, CureGRIN, for your support of this event, because I think without Calliope and Clarissa helping us, I don't think we would have, this conference would not come out so nice. But thank you very much. And I know the rest cannot applaud, but I'll do. Thank you.